In this lecture, we're going to look at how antiviral drugs work. I'll assume that you understand the basic viral life cycle. What we're going to do is to look through that virus life cycle and spot all the points at which we could use a drug, or we do use drugs, to stop the virus from doing what it wants to do. So let's start with here's our cell surface, and we have some sort of proteins or receptors on the cell surface, and along comes a virus. Now we're going to choose in this case a retrovirus. All sorts of jokes about retrovirus being old-fashioned or that kind of stuff, such as causes HIV. Right, so that's what we're going to be imagining. The retrovirus contains RNA. First thing the virus has to do, or I should probably call it the virion, shouldn't I really? The first thing it has to do is to attach to the cell receptors for it to become endocytosed. And we can actually interfere here, pharmacology, with drugs which stop this attachment. And one example of that is enfuvirtide. So I'm going to put the drugs in blue. Enfuvirtide. And that stops the fusion of the virus with the cell. Then the virus enters into the cell through endocytosis. Let's assume it's actually got past the enfuvirtide. So here's now it's inside the cell, and the coat is dissolved, and we've now got our RNA here. Because this is a retrovirus, it's somehow got to make DNA from that, and it usually does it through reverse transcriptase. So the reverse transcriptase tries to make DNA from the RNA, and we can inhibit this with reverse transcriptase inhibitors, or RTIs, such as zidovudine, which is also known as AZT, and is used in the treatment of AIDS. But there are other drugs that also inhibit reverse transcriptases, which are not just for AIDS. So carrying on our virus's story, let's assume that it's managed to evade the reverse transcriptase. Perhaps it's mutated and so it's resistant. It's now got to get its DNA into the host cell nucleus. So the DNA goes, remember the host has its own DNA. I'm going to draw this with a bit of a gap here, you'll see why in just a minute. So the viral newly synthesized DNA gets into the nucleus, and then it deploys an enzyme called an integrase. And what the integrase does is it integrates the viral DNA into the host genome effectively, merges it in, so effectively changes our genome. Right. So just make mine stop it coming out of the nucleus. So what we can do, of course, is to inhibit the integrase. And this is what raltegravir does. So if the virus has survived thus far, it then will be translating or using this DNA now to make its own new RNA, and it starts to assemble various proteins together, but it usually makes them as long polyproteins, and then it requires its own proteases. So these are viral proteases. So it's one of the things that it makes from its DNA. It makes these proteases, and that breaks these up into the component proteins. So you now have the proteins that it needs to actually make a new shell to encapsulate the DNA that it's made to make a new virus. So what we can do, of course, is to inhibit these proteases. So there's the basis of protease inhibitors. Such as duranavir. So let's imagine that this virus has got past even all these defences, then it needs to actually get it's made its new viruses, and these viruses want to leave the cell, and so they do that through a process that hijacks the endocytotic machinery. So what happens is the virion has on the surface molecules of hemagglutinin. So on the surface of the host cell you have receptors containing, well, a certain type of receptor containing salicylic acid. It's not important. And what happens is that when the virus gets, or the virion gets exocytosed, part of that process involves it being attached to these receptors. And this attachment has to be cut, and it's done by neuraminidase. So what we do is we try to stop this by using neuraminidase inhibitors. One example of this is a drug called oseltamivir, which we might know as Tamiflu, which was stockpiled by governments a little bit recently. So that then is our survey of the way in which antiviral drugs work. That's it.